Greetings to all. We are from the research team of Professor Wang Yiming at Nanjing Agricultural University. It is our great honor to introduce to you a study titled Immune-enriched Phyllosphere microbiome and rice panicle exhibits protective effects against rice blast and rice false smut diseases, which has been recently published in the journal iMeta. Rice serves as a primary food source for hundreds of millions of people worldwide. However, pests and diseases cause incalculable losses to rice yields every year. Despite the widespread use of chemical pesticides, growers still suffer losses of 10% to 23% of their grain production annually. Moreover, the extensive use of chemical pesticides poses irreversible hazards to human health and the environment. Beneficial microorganisms have shown positive effects not only in plants but also in animal and human health, providing possibilities for using beneficial microorganisms to improve crop health. Our research indicates that fungal pathogens trigger plant immunity upon rice panicle infection, altering associated microbiota composition. The enriched microorganisms from this immune response inhibit fungal pathogen growth. We developed a simplified synthetic microbial community that boosts rice resistance to fungal pathogens when used in the field. At the rice panicle initiation stage, we injected either sterile water or a spore suspension of blast and false smut into the panicles, followed by tissue sampling 48 hours post-injection for analysis. Through 16S RNA sequencing and classification, we identified a panicle microbiota comprising 6 phyla, 51 genera, and 137 OTUs. PC revealed distinct clusters between control and fungal infected panicles, signifying pathogen-induced shifts in the panicle-associated microbial community. Post-infection, alpha diversity analysis indicated reduced bacterial diversity, with false smut having a more pronounced effect than blast. Conversely, microbial abundance increased. Volcano plot analysis identified differential bacterial community accumulation post-infection relative to controls. Blast infection significantly enriched 5 OTUs from Acidobacter, Pseudomonas, Sphingomonas, and Rhizobium, while decreasing 11 OTUs. False smut infection enriched 3 OTUs from Burkholderia and Xanthoma nudacea, decreasing 17 OTUs. To further understand the changes in microorganisms after fungal pathogen infection, linear discriminant analysis was performed, and the results showed that after blast fungus infection, Pseudomonas and Sphingomonas were specifically enriched, while after false smut fungus infection, Dacheng and Intrabacter were specifically enriched. During pathogen infection, rice recruits beneficial bacteria to counteract invasion. From panicle samples, 189 cultivable bacteria were isolated, with 11 types selected from differentially enriched communities post-blast or false smut infection, including Pseudomonas, Acidobacter, Burkholderia, Delphtia, Methylobacterium, and Sphingomonas. In vitro assays demonstrated strong antagonism by Pseudomonas, Acidobacter, Burkholderia, and others against blast and false smut fungi, while decreased OTUs post-infection lacked inhibitory effects. Selected from plate antagonism assays for their strong pathogen inhibition, 3 bacterial strains, Pseudomonas, Acidobacter, and Burkholderia, were combined to form a synthetic community, Synchum, applied to rice leaves in various combinations. These were followed by blast inoculation after 24 hours. Results showed individual strains slightly improved resistance. Dual combinations enhanced it significantly, and the Synchum blend provided superior control against rice blast and false smut disease compared to single or paired strains. Field trials of Synchum on rice across seedling transplanting, tillering, and panicle emergence stages revealed that treated plants had significantly higher resistance to false smut and panicle neck blast compared to untreated ones, with concurrent increases in rice yield. Data suggests that Synchum application boosts both yield and resistance to these diseases. In summary, in this study, we found that activating immune responses can enhance the richness of beneficial microbial communities within rice panicles. The immune-activated enrichment of panicle-associated microorganisms shows inhibitory effects on fungal pathogens. Moreover, the synthetic microbial community enhances rice resistance to fungal pathogens in the field environment.